And yet, for a long time, it's been fairly well known that oils or liquid fatty substances are probably better for us health-wise. So, if you take what is an oil and only convert it partway to a fat through hydrogenation, perhaps you have something that's better for people than a fat. So, what do? Take this polyunsaturated oil, make it partially saturated, but don't go all the way too saturated, and you have something that can be used as a semi-solid fatty substance. Margarines and spreads are produced by what's called the partial hydrogenation of polyunsaturated vegetable oils. In fact, go home, if you have some in your refrigerator, look at margarine, look at soft spreads, and you'll find the words partially hydrogenated on their labels. There is a little bit of a problem with this process, or there was anyway. Partial hydrogenation was found to produce what are called trans fats. This was a complication of the technique they were using to partially hydrogenate the oils. In oils, there are double bonds, and they take a configuration or a geometry that's called cis. That seems to be the most natural way. But what happens is, during certain types of hydrogenation reactions, the cis bonds open, rotate, but don't undergo hydrogenation, and close, but in the opposite geometry. They now have a trans geometry. This creates what are called trans fats. Nobody thought that was a very big problem for a while, but gradually it became obvious it was a problem. They have now figured out hydrogenation processes that do not create these trans fats. Just to get a look at what that might look like, here would be the hydrocarbon end of a fat or a fatty acid. And this would be if it was saturated. You've seen this zigzag over and over again. Here's two forms of double bond. This is what's called a cis geometry double bond, and this is a trans geometry double bond. Notice that they have very different shapes, and if you have a pretty good visual eye, you'll see that this shape is an awful lot like that shape. And that actually is where the problem lies.
So, since naturally occurring oils contain almost exclusively cis carbon-to-carbon double bonds, and a complication of using partial hydrogenation of oils produces some trans carbon-to-carbon double bonds, this tendency of the natural cis double bonds to open, not undergo hydrogenation, and then reform as trans double bonds causes some serious health issues. They are now using different methods and more expensive catalysts to change this to a more favorable amount of hydrogenation without introducing carbon-to-carbon trans double bond.
You can get a look at this. If we just move this trans double bond on top of the saturated compound, except for the double bond, you can't even tell that they're different. And so this is a problem. Trans fats are a lot like saturated fats, and so they've been removed from most foods. Here's another example of an addition reaction application, the iodine test for unsaturation in foods. Foods can be tested with an I2 solution. It has a color, and if the iodine, the I2 molecule, comes apart, splits, and bonds to the two carbons of a double bond, the color goes away. And so actually watching the change in the color can tell you when you finally saturated all of the double bonds in the compound. And so the more X2 or I2 that can be added, the more unsaturated the compound is. So this can be used on a mixture of materials that might be going to be eaten just to get an idea, a rough measure of the amount of unsaturation. Oils, since they have a lot more double bonds in them, will be able to add or react with more iodine. They actually have this thing called an iodine number that's assigned based on this experiment. Oils have iodine numbers that are large, over 70, and fats have little or no iodine that they can add, and so they have iodine numbers less than 70. It has to do with the presence or absence of double bonds.
So here's one more application of addition reactions to carbon-carbon double bonds. Iodine has long been known as a good antiseptic, but getting it into deep, open wounds of large animals has been problematic. There's an older use in veterinary medicine of taking the compound pinene, which is in turpentine, and reacting it with iodine.
In the past, as a form of antisepsis, veterinarians used the reactions of iodine with turpentine, which is 60% alpha-pinene, to force iodine into deep wounds of large animals.
This should then drive iodine deep into the wound and get the antiseptic material in contact with the bacteria it's supposed to kill. Obviously, this would be pretty traumatic for the animal, but sepsis would be fatal.
Another type of reaction is condensation reaction. Condensation reactions involve joining or condensing together two smaller compounds into one larger compound with the release of a small stable molecule such as water. Sometimes this is called dehydration synthesis, to make something by removing water. It's called esterification if an ester is the product, and it's called amidation or amidization if an amide is the product. They could also just be called ester formation and amide formation. They're both condensation reactions, but they produce different products. Esterification, or the reaction to make an ester, involves a carboxylic acid, an alcohol, and the products are an ester and water. This was a reaction that you did in the lab. You mixed various carboxylic acids with various alcohols, and you got specific esters, and water was one of the byproducts. Here's a very specific one. Here's our carboxylic acid. Here's the alcohol. Here's an ester, C double bond O. Notice that this portion of the alcohol has become this portion of the ester, and here's what used to be the carboxylic acid. And we get water as another product. The names for all of these things would be acetic acid, ethanol, water, and then this particular ester is called ethyl ethanoate or ethyl acetate. This is a very familiar substance to a lot of people. Ethyl acetate smells like nail polish remover. It's one of the main components of nail polish remover. It may be one of the least pleasant smelling esters that there is, but it does still have a more pleasant smell than many compounds. Here's another esterification. Again, we have a carboxylic acid, an alcohol, making an ester, C double bond OC, and water. These particular ones are propanoic acid, a 3-carbon acid, methanol, a 1-carbon alcohol. They make methyl propanoate and water. Methyl propanoate is also another fairly common ester, and it has the odor of rum. Very fruity, very pleasant odor. Fats and oils are also esters. They're triesters of the alcohol glycerol. They're formed by the condensation of 3 separate fatty acids with the 3 alcohol groups on a glycerol molecule. So if you take some fatty acids and glycerol, you get either a fat or an oil, and then some water is also produced. So here would be glycerol. Notice it has 3 alcohol groups. Here's a fatty acid, C double bond OH, in a long hydrocarbon chain. C double bond OH, C double bond OH, long hydrocarbon chains on all of those. If those are allowed to react, they'll form a fat where we have an ester, and an ester. This is a triester of the alcohol glycerol. This particular one really will be a fat because these are saturated hydrocarbon chains, and that makes a solid substance which we recognize as a fat. Amidation, or the formation of an amide, takes a carboxylic acid plus an amine to make an amide and water. So here's our carboxylic acid, here's our amine, and here's our amide, C double bond O, single bond N, has merged this compound with this, and we've lost an OH and an H, and that's where we got the water. The names happen to be butanoic acid, a 4-carbon carboxylic acid, methylamine, and methylbutanamide. Butanamide because it's a 4-carbon amide, and N-methyl to tell us, hey, that nitrogen has a methyl group on it. And that's the result of that amide reaction. Peptides and proteins are also amides. They're formed by condensation of specific amino acids in a specific order. For example, take the amino acid valine, notice the amino and carboxylic acid group, and the amino acid glycine, amino and carboxylic acid group. What makes them different is this side group. And they'll condense together, the carboxylic acid end of one and the amino end of another, to make an amide. Here it is right here. See double bond O, single bond N. We're still left with an amino residue and a carboxylic acid residue. This little thing is called a dipeptide. It's 2 amino acids linked together by 1 amide bond. Combustion is a reaction with oxygen, burning in the presence of oxygen, releasing energy. Usually it's an organic compound that reacts with oxygen to produce carbon dioxide or carbon monoxide, water, and energy. We can have complete combustions, where the organic compound reacts with enough oxygen to turn all the carbons of the organic compound into carbon dioxide, steam, or water, and energy. Sometimes the combustion is incomplete. Generally, this means there's a deficiency of oxygen. The organic compound reacts as completely as it can with the oxygen available, but there isn't enough oxygen to make carbon dioxide, so carbon monoxide is formed instead, and steam and energy.
In just a little bit, we'll look at organic oxidation reactions that are not combustions. These are generally oxidations that happen to alcohols and aldehydes. Organic oxidation basically means looking for an increase in the number of bonds between carbon and oxygen within the compound. You have done or will do lots of different organic reactions in your labs. You'll have done condensation to make nylon. That was an amine reacting with an acid chloride, a substitute for a carboxylic acid. That was amidation. To make esters, you also took alcohols and reacted them with carboxylic acids. That's esterification. You made 2,4-DMP derivatives. That actually turns out to be a condensation reaction. You're going to make aspirin. That's an alcohol reacting with a carboxylic acid, although we'll use the anhydride of the carboxylic acid. It's another esterification. You'll also do some organic oxidation reactions. You'll burn up some hydrocarbons and other organic compounds to see combustion reactions in action. You'll also try oxidizing some alcohols and aldehydes with oxidizing agents like KMNO4 and Na2Cr2O7. You'll also test the difference between sugars, whether they're oxidizable or not, with Benedict's reagent. Remember that your lab book is a source of a lot of information about these various reactions. There are samples there, and there are written introductions, much like a textbook, concerning each of the reactions.
You have a worksheet in your packet called Classifying Organic Compounds and Reactions Worksheet. Use it in several ways. One thing to do is to take all the organic compounds and put them in their organic families by their functional group, and then label each reaction as one of the 4 organic reaction types that we were looking at: combustion, addition, esterification, or amidation. If it's a combustion, try to go farther, complete or incomplete. And if it's an addition, see if you can apply one of these other words: hydrogenation, hydration, and halogenation. Most reactions require some sort of additional reagents, some reaction conditions that are controlled, like temperature or solvent, or some specific catalyst to initiate the change. Don't really worry about whether those are shown or not shown at this point. Assume that every combination in these organic reaction possibilities will be a reaction, and use that sheet to practice. You can find a complete key online for this worksheet. A brief key follows.